Hello friends, welcome back to my course on microbial biotechnology. We are in module 9 discussing about food production involving microorganisms and their products. So today we are going to learn about single cell protein production, which we shortly call as SCP. So this has broadly three sections. The first section deals with the introduction and overview of single cell protein. The second section deals with the SCP production and the key factors. The third section deals with the applications, impact and future of SCP. So, in the first section we are going to learn about the introduction, importance of SCP, historical development of SCP, sources of SCP, merits and demerits of single cell protein, SCP from different microorganisms. So, let us learn about what is the challenge we are really trying to address in this particular lecture. We know that our population globally is increasing rapidly, and this has resulted in the increased demand for protein, and this is expected to rise by around 40% by the year 2050. This rising demand poses a significant challenge for meeting our nutritional needs. 40% increase in protein means we need 40% additional resources to produce them, maybe a little bit more resources to produce them in terms of land, water and other inputs. Contributing to this challenge is the shrinking availability of land and water per person due to climate change and unplanned developmental activities. And we know that urbanization is also rapidly increasing, thereby reducing the cultivable land. These factors are reducing the amount of arable land available for growing crops and raising livestock, which actually provides us a protein. Now, this is the trend in per capita protein consumption from 1961 to 2011. So, here we see the blue color one is the global average, and then you see the green one are the consumption pattern in the developing world, and the red one is the consumption pattern in the developed world. In both the two different economies, the consumption is actually increasing every year or every decade. So, and it is likely to grow and reach additional 40% by 2050, as we have already discussed. So, to produce this has become a big challenge because our natural resources are depleting and waste generation is increasing. Together, these issues complicate the process of producing enough protein to satisfy the growing demand. In this context, ensuring nutritional security becomes a pressing concern as we seek solutions to address these interconnected challenges effectively. One idea could be whether we can take these waste, which is right now a challenge, as actually a resource in our scheme of things and use them for protein production. So, these you can see children playing in a garbage dump in Africa, just to represent the menace of growing urban wastage, or even in the rural areas now wastage is becoming very omnipresent. Particularly, we have the plastic wastes which are not degradable and they are just floating around in the environment. So, what is the solution against this protein demand, increased protein demand in a backdrop of decreased natural resources like land, water and other inputs? Single cell protein is a promising alternative that can support both human and animal nutrition in such a scenario, and we can actually use the agricultural wastes, and then these can be used for production of microorganisms which can produce single cell proteins that can be used for animal feed as well as human food, and of course, subject to consumers' attitude and acceptance, and packaging is going to play a big role over here, and including the processing part. So, we look into these disadvantages as a kind of an advantage whereby we can meet the nutritional demand of proteins by using agricultural waste and producing protein out of that. So, we have efficient microbial single cell producers and production systems which can utilize solid and liquid agricultural waste and convert them into resources. Let us have an overview of microbial process applications, highlighting state-of-the-art methods and the role of genetic engineering in the current and future enhancements. So, here we have the traditional fermentation, and then the basically state-of-the-art. It has been used historically and for long, and currently in processes like alcohol production, food preservation, taste and texture development. Then from the engineering and future developments perspective, active preservation of the traditional techniques, historical research into understanding early fermentation techniques in ancient cultures is very, very relevant. Now, we have methods by which we have enhanced these traditional fermentation, where we have gone for strain selection and then bred or engineered to improve the taste, texture and the health benefits. And for these we use omics tools to select and breed optimal strains for processes. Engineered strains producing additional vitamins, then PUFAs, taste, odor in fermenting strains. Then in the case of animal feed, biomass are used in feed as nutritional supplement, which is a primary food source, or as nutraceutical and or therapeutic platform. Here strains chosen were engineered to match needs of specific animals, agricultural methods and aims. Engineered into a therapeutic platform, biomass growth on waste substrate to optimize sustainability. We also have a focus on ingredient production, where production and purification of native or recombinant food additives for nutrition, taste, odor and processing purposes. Here engineered pathways to produce non-indigenous chemicals, proteins or other biological substances are undertaken. Engineered living therapeutics drugs delivered using probiotics are also one of the focus. Then we have meat as well as the dairy alternatives. Microbial meat analogs recreating nutrition, taste and texture is one of the goals of these. The production of animal products components in cell factories is another. Here we go for engineered nutritional or texture elements, allowing for closer resemblance to meat, and then animal flavorings produced in cells to such as hemoglobin or lactose. Then the current topic of our discussion, single cell protein. Here microbial food product is included as a primary source of protein, and this is a complete food source and is used as a health supplement. Basically we go for engineered approaches to improve the nutritional profiles, then engineered into a therapeutic platform, growth on waste substrates to optimize sustainability, and then engineered taste compounds to improve organoleptic properties. So, in this scenario all of them are actually important. The traditional fermentation will continue, the enhanced fermentation as well as other approaches like the meat and dairy alternatives, but then single cell protein is also emerging as an important vertical in mitigating the nutritional demands of protein. Now, what is this single cell proteins? We can simply define it as something which refers to a biomass of dried cells produced from microorganisms, which may be algae, yeast, bacteria and fungi, and these are non-toxic. SCP is also known as a bioprotein, microbial protein or biomass. SCP serves as a protein rich supplement or ingredient for both human and animal diets. It is a valuable alternative to traditional protein sources, and apart from its use as human food, animal feed, it is finding wide applications as bioplastics and in enzyme production and wastewater treatment. Let us look into the protein supply chain, traditional versus the single protein production. So, we divide these into different discussion points like raw material and growth and cultivation, how we harvest and process, what are the quality control and safety standards in place, how do you distribute and store, and regarding the aspects of retail and customer. So, in the traditional protein production process, we go for livestock breeding or feed production, and then we go for rearing, and then harvesting and processing is done by slaughtering and butchering. Then we go for quality 6, antibiotics and hormone which is need to be monitored, and then we go for cold storage distribution, and this retailing and is done mostly through supermarkets, butcher shops, restaurants, etc. In the case of single cell protein production, the process is entirely different. We do not have livestock, here we have microbes. So, we go for strain selection and culture media preparation in place of feed production. Then instead of rearing, we go for fermentation, and there is no any slaughtering and butchering here. We just go for harvesting cells, of course refining them. Then of course, the quality control and nutrient content validation is important over here, and then we may require cold chain distribution, we may not require in certain cases, we can simply go for distribution. Initially these are specialized retailers or as an ingredient in the processed food, then mainstreaming outlets as the market would be growing. So, these are the two differences between the traditional versus single cell protein production. What are the key characteristics of single cell proteins? They are produced by diverse microorganisms like algae, yeast, bacteria and fungi. Single cell protein productions comes with a baggage of rather advantage points of like environmental benefits, because it uses less land and water than plant based protein sources and does not contribute to greenhouse gas emissions. Rather, some of the greenhouse gas gases can actually be converted into proteins, and that can be one of the steps in climate change mitigation. Then we have the advantage of year round production. In case of animals, there may be challenges, or in the case of crop based proteins, it may not be year round, but since we are growing these under artificial conditions and controlled conditions, we can go for year round productions. So, there is no any climatic constraints. So, and we get a continuous supply of single cell proteins. So, you now to some extent know what could be the importance of SCP. For example, it can be a tool for climate change mitigation. It can help us in converting agricultural waste into wealth, but let us try to learn them in a more structured way. Number one, it addresses the protein demand or the gap in that protein demand, because we know that there is a rising consumption globally of protein, and it is projected to increase from 40 kg per capita in 2013 to around 51.5 kg in 2050. And in this context, SCP offers a cost effective and nutritionally rich alternative to meet this demand. From a nutritional point of view, SCP can have many advantages, but it actually varies depending on the microorganism and the substrates used, including the amino acids, proteins, carbohydrates and lipids. SCP provides essential macro and micronutrients, including proteins, lipids, carbohydrates, vitamins and minerals. From the production point of view, and if you look into the production efficiency and the nutritional composition, SCP's protein content ranges from 30 to 70%. It can be enriched through genetic engineering to match the amino acid profile of traditional proteins like fish or soy protein, and it is environmentally sustainable. SCP can be produced from readily available and inexpensive raw materials such as carbon sources, example methanol, ethanol, renewable sources, example molasses, waste substances, agricultural and industrial, and even greenhouse gases, example methane. Let us have a small discussion on the history of the development of SCP. So, SCP was known as early as World War I, when SCP research actually began with initial methods developed in Denmark and Germany. In 1919, Zulauf-Verfahren method was developed using aerated yeast and sugar solutions, and in the Second World War, yeast such as Saccharomyces cerevisiae and Candida species were used for protein production. Then after the Second World War, in the post-war period from 1960s to 1970s, SCP industries expanded in the UK, France, Italy, Russia, Japan and Taiwan, and in 1966 the term single cell protein was coined by Carroll L. Winson. Then various technological advances has taken place over the time. For example, the British Petroleum developed proteins from the oil in the 1960s for production SCP from oil. Then the Soviet Union established large plants in the 1970s and 1980s, but it faced closure due to environmental issues. What are the sources of single cell proteins? We can have it from different microbes like bacteria, microalgae as well as fungi. The generation time in bacteria is very short, 20 to 120 minutes, allowing rapid biomass production. The substrates the bacteria use is various organic and petrochemical wastes, and some of the examples are Methylophilus species, which is notable for its fast growth and high quality protein. Microalgae like spirulina and chlorella offer high protein content up to 70% and are very commercially successful, and additional nutrients like omega 3 fatty acids, vitamins and minerals. Then from the cultivation point of view, it is efficient in utilizing solar energy and are simple to cultivate. In the case of fungi, they have 30 to 50% protein and provide essential B vitamins, and the Pekilo process in Finland uses Paecilomyces varioti for SCP production. So, microalgae we can see here around 60 to 70% protein, and these are mostly phototrophic production of omega 3 fatty acids, and it has certain of course disadvantages like economical limitations of scale up, then digestibility, a need of cell wall disruption to release the nutrients. Some of the examples are Tetraselmis sucica, then Isochrysis galbana, etc. Then in the case of yeast and fungi, we have roughly around yeast has 38 to 52% protein, fungi has 0.23 to 15% protein, and they use different substrates and they can produce vitamins and micronutrients. Some of the potential disadvantages are improvement of protein content and essential amino acid profile, possible presence of toxins as well. Saccharomyces cerevisiae, Cloviromyces marxianus are some of the yeast which are used for SCP production. And then in the case of fungi, we have the Aspergillus oryzae, Yaroia lipolytica, which produces SCP. Bacteria has around 50 to 80% protein content. It is a high protein content, and growth on C1 substrate is one of the characteristics. However, they have certain disadvantages like palatability issues, high content of nucleic acids, production of toxins. Some of the examples are Methylobacterium, Extroquens, then Methylococcus capsulatus, etc. What are the merits and demerits of single cell protein from different microorganisms which we just discussed? So, we will discuss these disadvantages and advantages with respect to each of these organisms one by one. So, in this picture you can see a colorized scanning electron micrograph of a bacteria, and some of the advantages of having SCP from bacteria is that we have diverse bacterial species which can be used for SCP production. Then these diverse species utilize diverse substrates, and therefore, we have many different options by putting a permutation and combination of these bacterial species and the available substrates. Certain bacteria like Methylophilus and Methylotrophus are used at the commercial scale. So, there is no any problem of scaling up, and we know bacteria has a very high SCP content and can be as high as 80% or 80% plus crude protein, and it has a good amino acid profile, though it may be low in sulfur containing amino acids. However, it has certain disadvantages like elevated nucleic acid content. Bacterial SCP may contain high levels of nucleic acids, and then maintaining sterility is crucial due to bacterial pH measurements, then there is a risk of contamination by pathogenic bacteria. And also there is a challenge of recovery of bacterial SCP, which can be actually difficult. Then there is always endotoxin production risks. So, it needs careful evaluation, especially with gram negative bacteria. Let us now look into the advantages of having single cell proteins from algae. It has very high content, around 60% crude protein. It is suitable for animal feed, and algae grow using carbon dioxide and sunlight, which are low cost resources. So, it basically uses cost effective substrates. However, there are certain disadvantages. For example, it has rich chlorophyll content, which is unsuitable for direct human consumption. Then there is a low cell density. Algae often have low cell density, yielding only 1 to 2 grams of dry weight per liter, and there is a contamination risk. Algae cultivation can be prone to contamination affecting the SCP quality, and the recovery methods for getting the unicellular algae can be very expensive. So, here you can see a right handed spirulina. Then what are the advantages of using filamentous fungi for producing single cell protein? So, it can be raised on diverse feedstock. Filamentous fungi can use various polysaccharide hydrolysates such as starch and sulfite liquor from wood pulp industries, and it has moderate crude protein content. SCP from filamentous fungi generally contains 50 to 55% crude protein. Recovery is straightforward, often achieved through filtration. And here you can see a fungal hypha and mycelium. However, there are many disadvantages of obtaining single cell proteins from filamentous fungi. For example, they grow very slowly compared to bacteria and yeast, and there is a higher risk of contamination by yeasts during production, and it has elevated nucleic acid content, which can impact SCP purity, and potential mycotoxin production requires evaluation for product safety. Let us look into the single cell protein production from yeast. It has advantages like it is a high crude protein content, typically around 50 to 60% crude protein. Then it has balanced amino acid comparison. Yeast SCP provide a well balanced amino acid profile, though it may lack sulfur containing amino acids. Yeast SCP is rich in vitamin B, and it is used in both human food and animal feed. There is a low bacterial contamination risk. Yeast SCP generally has a low risk of bacterial contamination. It is an easy recovery process. Recovery is facilitated by continuous centrifugation methods. However, it has many disadvantages like slower growth rate, then elevated nucleic acid content, deficiency in sulfur containing amino acids. SCP may lack these essential amino acids in many instances. This is the yeast cells in gram staining of Candida growth on SDA. Now, let us see this figure, which shows in A the components of microorganisms beneficial for nutrition, including proteins, fiber and bioactive compounds. So, we have high protein content in most microorganisms, then they grow on waste substances, and it produce vitamins in many microorganisms. Then nutritional chemicals such as phytochemicals and antioxidants are also produced, and it has high fiber in mycelium and yeast strains, which include fibers such as beta glucan. So, now if you compare the levels of fiber, protein and micronutrients in SCP of various microorganisms used in food applications, you can see in figure B with these spots, which is proportional. So, in bacteria the fiber is very low, protein is quite good, micronutrients also satisfactory, but algae fiber is very high, protein is comparatively lesser. So, in the case of mycelium and yeast when compared to bacteria, but algae provides a lot of micronutrients amongst all these various microorganisms, and so algae, mycelium and yeast actually provides a lot of fiber. So, this is a pictorial representation of the different components and produced by the different microorganisms. Let us now move to section 2, where we will be discussing about the key factors and production process of single cell proteins. So, here we will go for the production stages, optimization of SCP production, key considerations for selecting microorganisms for SCP production, the critical properties of SCP, then production process, factors affecting SCP production, substrate requirements for SCP production using agricultural waste and byproduct streams. So, SCP production can be divided into 3 distinct stages, like the upstream process, which is the initial stage, and it is a preparative stage where sterilized raw materials are introduced into the reactor and microorganisms are added to this sterilized raw material kept in a controlled environment. In the midstream process, the focus shifts to growing these microorganisms on this substrate. This stage is where we produce a large amount of biomass rich in protein. And in the downstream process, which is the final stage, we harvest the biomass and turn it into a usable product. This involves collecting the microorganisms and their products and processing them into forms that can be used as food supplements or other products. So, how do you optimize these entire SCP production? It begins with the selection of the microorganism, which we need to choose for optimal growth, productivity and substrate utilization. Then we go for optimizing the cultural conditions like temperature, pH and nutrient levels. Then before we introduce the substrate, there is a pre-treatment whereby we enhance the nutrient availability. Then we go for nutrient supplementation to ensure balanced nutrient supply, and selection of the fermentation process is very important, which needs to be based on the microorganism and the substrate characteristics. State improvement is one of the important thing, where techniques like mutation breeding and genetic engineering for enhanced traits are used, and here we can see optimal single cell protein production process, which integrates the circular economy approaches. So, here we have 2 type of waste which can serve as substrates, the agricultural and food waste and urban waste, and this we need to go for sterilization, and we grow at a laboratory scale different organisms like microalgae, yeast and fungi and bacteria, and these producers of SCP are acquired from collections or they are isolated from natural habitat. When you mean acquisition from collections, it can be also the various government or private repositories. And for the substrate selection, we have to be careful that the substrate is non-toxic, it is fully regenerable, contains a carbon source and capable of supporting multiplication of microorganisms, and the substrate is economic and not that costly. And after we do the SCP production optimization at the laboratory scale, we go for scaling up at the pilot scale, where we go for production of microbial biomass and the metabolites, and we go for the evaluation of nutritional and toxicological aspects and the reproduction rates and recovery levels. And after optimization and standardization of the production, we go for further scaling up at the industrial level production. So, here we go for cell wall disruption, filtration, concentration, purification, and then of course, elimination of toxic compounds and some nucleic acids to enhance the protein percentage, and we get the final product, and this can be used as a animal feed or as a human food. So, this is the optimal single cell protein production process, starting from the laboratory scale to the pilot scale to the industrial or commercial scale. So, one of the important thing in SCP production is selection of the microorganisms. So, When selecting microorganisms for single cell production from methane, for example, several key priority properties are critical to ensure that the final product is safe, nutritious and commercially viable. Microorganisms used in SCP production from methane should be safe, produce high quality and digestible protein, grow rapidly, adapt to various environmental conditions and efficiently utilize methane and nitrogen sources. These properties ensure that the SCP is not only effective as a protein source, but also economically viable and safe for its intended applications. What are the advantages and disadvantages of single cell protein production? So, we already know that for SCP, SCP is actually a solution to the challenges of scarcity of land and water resources and other resources. So, advantage is that it requires very less reduced land and reduced resource, and then low water usage, lower greenhouse gas emissions, it causes less pollution, year round and efficient production, reduced overfishing, nutritional cycling, because we are producing the protein in the artificial conditions and we are not going for harvesting the fish either in the rivers, wetlands or in the oceans. And because we are using the agricultural waste or some urban waste, we are recycling the nutrient. And because we are using less land, there is less land degradation, and then this results in resource efficiency and reduced biodiversity loss, and it ensures food security. However, there are certain disadvantages because we may not be using the land and other resources, but we require a lot of energy. So, there is a high energy consumption and there is a resource competition sometimes. Biomass separation is a challenge, separating the protein content from the total cell mass. Then there is a dependency on raw materials at all levels. Even the waste can be a limiting factor sometimes, which is used as a substrate. A potential limited amino acid profile and scale up challenges are there. There are also challenges of new type of waste generation, and external cooling systems are required, and this is very capital intensive. So, what are the critical properties of SCP? Number one is absence of pathogenicity and toxicity. Microorganisms for SCP production must be non-pathogenic, ensuring they do not cause disease or health issues, especially if used as animal feed or human feed. The microorganisms must not produce harmful metabolites or toxins. So, absence of toxicity is very important, and the substrates should also not be toxic in large amounts or accumulate in the final product. The protein content and quality, the microorganism should produce a high protein content relative to its biomass, measured as a percentage of its dry weight to ensure SCP is a valuable nutritional source. The quality of the protein is also important and is determined by amino acid profile. The SCP should offer a balanced profile of essential amino acids, ideally surpassing traditional protein sources like soy or meat to meet human and animal nutritional needs. Digestibility and organoleptic qualities of the SCP are very important. It should be easily digestible by the target organisms, whether animals or humans. This involves not only the protein content, but also how well it can be broken down and utilized by the digestive system of these organisms. Organoleptic qualities for SCP used in food applications. Organoleptic qualities such as taste, odor and texture are important. The SCP should be palatable and not impart any off flavors or undesirable textures that might affect its acceptability. The next important property is the growth rate. The microorganisms should have a high growth rate, which translates to efficient production of SCP. Fast growing microorganisms can reduce production time and costs, making the process more economically viable. In addition to the growth rate, the productivity of SCP production is a crucial factor. High productivity ensures that the bioprocess is cost effective and efficient. Another important property is the adaptability to environmental conditions. For example, pH, the microorganisms should grow well across various pH conditions, or at least within easily controlled pH levels in the bioreactor. Adaptability to acidic or alkaline environment is important for optimizing growth. Next important parameter is temperature. Microorganisms have varying temperature needs. The Susan microorganisms should thrive within an optimal temperature range and tolerate fluctuations or deviations from it. Then another important factor is the concentration of the minerals. The microorganisms should be able to thrive in the presence of varying concentrations of minerals and other nutrients. This includes the ability to adapt to potentially limiting or excessive levels of trace elements or salts in the growth medium. Another important property is the ability to utilize carbon and nitrogen sources. For example, the microorganism must efficiently utilize methane or other carbon sources for growth, possessing the metabolic pathways and enzymes needed to convert methane into cellular biomass effectively. SCP production requires sufficient nitrogen for protein production or protein synthesis. The microorganisms should efficiently use nitrogen sources like ammonia, urea or other compounds, as nitrogen is essential for producing amino acids. Let us now have a discussion on the general production process, some of which are actually known to you from earlier discussions. So, substrate and strain selection is very, very important, as we already know, but the criteria is selecting fast growing microorganisms which gives high protein content and have suitable growth characteristics. Then substrate preparation includes pre-treatment methods like grinding, sterilization and hydrolysis. Then the fermentation technology is also very, very important. We may go for submerged fermentation, which utilizes liquid growth medium, often continuous with biomass, required through filtration or centrifugation, or we may go for semi-solid fermentation, which involves solid or semi-solid substrates with lower capital and operational costs, or we can go for solid state fermentation, which uses solid substrates incubated to produce SCP. And then the downstream processing like harvesting techniques, which include centrifugation, filtration or sedimentation, and these cell disruption methods like homogenization or chemical lysis to release the single cell protein, followed by purification for removing impurities using filtration, chromatography or precipitation, and finally the drying methods like freeze drying or spray drying. So, this is the schematics for commercial single cell protein production process. We begin with the strain selection and also the strain development. Then we go for inoculum in culture flask and growth optimization. Then the medium preparation, nutrients for growth with C1 carbon as carbon and energy source, and we go for the fermentation. After the fermentation is over, we go for harvesting, which is the collection of the cells produced by this fermentation process, and then we go for separation of cells from media through downstream process, which includes cell disruption and protein recovery, and then drying the cells or protein extract by spray drying, and look for the various quality parameters in the quality control step, and then finally once the quality is okay, we go for packaging, and then finally the product distribution. What are the factors which affect SCP production? The microorganism selection is the first important thing, and the growth rate, tolerance and substrate utilization are very, very important. Growth rate is essential for efficient production. Tolerance includes the tolerance against temperature fluctuations, either low or high, similarly low or high pH and other stress factors. Then efficiency of the low cost substrate is very, very crucial. The safety and acceptability is very, very important. The health risks include potential allergic reactions and nucleic acid content of the SCP final product. Then regulatory compliance is necessary for safe human or animal consumption. Let us now look into the substrate requirement for SCP production. The substrate must be non-toxic, renewable, cheap, non-seasonal and non-exotic. We try to get local waste resources, and We go for common substrates which include carbohydrates, fruit, molasses, starch, vegetable waste, natural gas, methanol, ethanol, petroleum byproducts and lignocellulosic biomass. Selection depends on availability and proximity to the production plant. If they are very far away, the transportation cost will build up into the cost of production. Let us now look into the potential substrates used for single cell protein production, which may include molasses, which are the byproducts of sugarcane and beet processing. These are rich in sugar, 40 to 55% and easy for microbial cultivation. Or we may go for dairy wastes, which are high in biological and chemical oxygen demand, contains fats, oils, phosphorus and nitrogen compounds. Or we may go for using fruit waste, which are rich in carbohydrates and nutrients for microbial growth, especially whole fruits and peels. Then we have starch rich sources and bran, tuber and grain residues rich in starch. Then bran contains starch, protein, rich fiber and nutrients. Then we may have soybean meal, which are high in protein, carbohydrates and minerals, and these are widely used in animal feed. And we may use methanol, for example, which are used by Imperial Chemical for SCP production. Then we may have waste from dates or plantation, palm plantation, which are rich in sugar, 60 to 70% used in fermentation, particularly the date syrup. And we may use agricultural wastes for SCP production. These includes rice bran, pineapple peels, brewers waste and sawdust. Agricultural waste is cheap, easily obtainable and supports microbial growth. So, let us look into the SCP production using agricultural waste and byproduct streams. Agricultural industrial waste are increasingly used for SCP production, ethanol and biogas, preventing environmental harm and adding economic value. Though costly initially, this approach is gaining importance due to future food supply concerns. Then we have the industrial waste, which includes paper and pulp effluents, methanol, oil, latex waste, crude glycerol, waste cooking oil, fruit waste, leaf juice and poultry and slaughterhouse waste. And here we can use different microbes like Cloveromyces, Candida, Saccharomyces, Pichia and so on. So, these are some of the rotten fruits and vegetables waste which we can use for SCP production. So, this figure we can see the renewable resources for SCP production, basically crop waste, straws, hulls, husk and bran, then combined agricultural waste, or we may have glycerol, which is a waste of many industrial processes, then fruit waste, peels, pulp, whole fruit and then molasses and then dairy wastes, which we have already discussed in the past few slides. Now, we can also use gasified waste as carbon source for SCP production. To enhance sustainability, the goal is to use gasified waste resources like methane, carbon monoxide and carbon dioxide as carbon source for SCP production. This approach supports a circular economy by recycling carbon waste and reducing reliance on fossil fuel. So, in this picture we have gasified waste resources as carbon sources for SCP production, support and transition to a circular economy. So, here we go for the gasification of the waste, and then we have intensified food production which may produce many of these waste due to food processing process. These are all gasified, and then these are fermented to create engineered single cell proteins. So, we get single cell proteins from fermentation of these gasified waste, which can be used as animal feed, and these animal feed can be resulting in enhanced growth of the animals, which can be source of proteins. And of course, the waste of these animal can be used back as fertilizer into the plant system. So, thereby this closes the loop. So, basically using agricultural waste products, gasifying them and using that gas for creating single cell protein by fermentation and using that as a food source for the animal, and the animal meat is used as the protein source, waste as a input back into the plant production system. So, now synthetic biology is advancing functional food production through precision fermentation with tools to engineer acetogens, methanotropes and hydrogenotropes. Many companies are leveraging these innovations to develop sustainable foods. So, here in this figure we have these gas fermentation with different gaseous products like methane, carbon dioxide, carbon monoxide, hydrogen and oxygen, and then this gasified waste is used as carbon feedstock for single cell protein production, which supports the carbon recycling in a circular economy. And you can see the movement of these carbon to various metabolic pathways in various organisms, which may be methanotropes, or which may be acetogens, or which may be hydrogenotropes. So, let us go for a little bit of discussion on the methane based SCP production technology. This is a promising technology because methane is a renewable and cost effective substrate. However, its low solubility presents challenges for efficient SCP production. Researchers have developed an innovative bioreactor using a hollow fiber membrane for methane delivery, achieving high SCP production with yields of 1.36 gram SCP per gram of methane, or 5.5 gram of SCP per gram of nitrogen, 67% protein content and over 42% essential amino acids. Additionally, the methane and ammonium were nearly fully utilized, showcasing the bioreactor's efficiency. So, here this is a schematic diagram of the SCP producing bioreactor that comprises three major components: the main reactor, the methane supply system, and the oxygen supply system. So, here is the suspended growth bioreactor, where single cell proteins are produced that can be used as a animal feed or as a fish feed. So, here you can see some anaerobic digestion taking place, which produces this methane, and these methane is used in these bioreactor. Let us now go to section 3, where we will be discussing about the applications, impact and future of single cell production. So, we will discuss about the commercial applications, then functional properties, nutritional profile and safety, sensorial aspects and consumer perception, economic and environmental considerations, emerging technologies for SCP production, and the future prospects. So, let us begin with the SCP in commercial applications. SCP provides an alternative to traditional protein sources like fish and soy, offering proteins, carbohydrates, nucleic acids, lipids, minerals, vitamins and essential amino acids. And it can all be also used to produce bioplastics, offering a sustainable alternative to conventional plastics, as well as SCP's amino acids and fatty acids can be used in enzyme synthesis. SCP can enhance microbial activity in wastewater treatment, aiding in organic matter degradation. So, this is the market revenue share of single cell protein by application in 2022. So, food and beverage is the major revenue generator, where 75% of the revenue comes from SCP utilization in the food and beverage sector, followed by its use as animal feed 15%, and then its use as dietary supplements 5%, and miscellaneous others another 5%. So, these are some of the companies and their products which are produced using various organisms, and their country of origin or production you can see, and the protein content of this product, and The product name you can see. Here for example, you have Prototiton, which is a trade name. It is produced using Clostridium species, the company called Biotech in USA, and it has around 85% content, protein content, and it is used produced in a ethanol plant, and it is used as salmon and shrimp feed. Then you have this Cyanotex spirulina, which is produced by using Arthospira platensis by this Cyanotex Corporation in USA, has around 60% protein content, and it is produced in deep ocean water, and this is one of the most commercialized SCP products. Then we have Linsight Nutri, produced using Saccharomyces cerevisiae by the company Laissefray in USA, having a content of 55.7% protein. It is produced by extrusion, and the product is available as dried inactive yeast. Then we have the Silpro, produced with the help of Candida utilis by the company R-Biome in USA, has a content of 60% protein, and it is basically a forestry byproduct, and the protein is comparable to soybean. Then we have Uniprotein, produced with the help of Methylococcus capsulatus by Unibio AS in Denmark, and has 70% protein content, produced using natural gas, and it has a particle size of around 150 to 200 micrometer. And there is Profloc, produced using bacteria by a company called Nutrinsic in USA, which has a 60% protein content, produced from wastewater from a local brewery, and it is used in fish meals in feeds. So, what are the functional properties of SCP? The functional properties of proteins such as solubility, emulsification, foaming and gelation depend on their origin and structure. Proteins are amphiphilic, interacting with both hydrophilic and hydrophobic environments. Solubility is crucial for their use in food systems and is influenced by pH. Higher pH values than a protein's isoelectric point improve solubility and stability. So, this table we have Methylobacterium species, which are known for their ability to metabolize C1 carbon compounds like methanol and methane, with protein content ranging from 50 to 80% depending on the strain. The most common species used for single cell protein production includes Methylophilus, Methylotrophus and Methylococcus capsulatus, and you can see these Methylotrophus has a very high crude protein content, 81 percentage, and the others followed by Capsulatus but 74 and 63 respectively. Proteins in emulsions stabilize the mixture with SCP from kefir microorganisms showing emulsifying properties similar to defeated soy flour. For foaming, proteins must stabilize foams effectively across various pH levels and concentrations. For example, protein PAO5 from Saccharomyces cerevisiae demonstrates notable foaming stabilizing properties. Gels formed by proteins or high molecular weight substances create a network that holds liquid. SCP gels have been found to be stronger than those made from soy flour. SCP's nutritional value is determined by its chemical composition, including amino acids, nucleic acids, minerals, enzymes and vitamins, alongside its high protein content, which is cost effective compared to plant and animal sources. Protein content, for example, dried cells of Pseudomonas species grown on petroleum based liquid paraffin can contain up to 69% protein. Algae derived SCP has around 40% protein. Microbial proteins contain all essential amino acids depending on the substrates and microorganisms used. Then we have amino acid profile. Bacterial SCP's amino acid profile resembles the fish protein. Yeast derived SCP is similar to soy protein. SCP is generally deficient in sulfur containing amino acids, methionine and cysteine, but rich in lysine and other amino acids, necessitating supplementation. SCP is rich in vitamins, especially vitamin B12 and those produced by bacteria, and vitamin A by algae. Common vitamins include riboflavin, thiamine, pyridoxine, niacin and others. Microorganisms grow quickly, yielding sufficient biomass. Algae in 3 to 6 hours, bacteria 30 minutes to 2 hours, yeast 40 minutes to 3 hours. SCP can be produced year round, independent of environmental conditions with low water requirements. When it comes to nutritional safety, SCP is a nutritious protein source with essential amino acids produced by microorganisms, offering alternatives to fish meal and soybean meal. Saccharomyces cerevisiae, for example, grown on sugar-based substrates, provides a broad amino acid range but lack threonine and tryptophan. Photohydrogenotropic bacteria, algae like Chlorella vulgaris and Arthrospira platensis have amino acid profiles similar to soybean meal, suitable for animal feed. Microbial biomass from algae and bacteria recovers nutrients from wastewater and produces SCP. However, some SCP sources have high nucleic acid content, posing health risk such as gout and kidney stones. For human consumption, nucleic acid levels must be below 2%. SCP also carries risk of allergies, toxicity and contamination. Let us look into the sensorial aspects and consumer perception. Interest in plant-based proteins, insect proteins, cultured meat and single-cell proteins is increasing, with plant-based proteins being the most accepted. SCP ranks second in acceptance, followed by insect proteins and cultured meat, which are often rejected due to neophobia and perceived unnaturalness. Cultural factors heavily influence the acceptance of alternative proteins. Research on SCP in food shows it can be added to bread, cereal bars and pasta without affecting sensory properties. Bread with up to 4% SCP remains acceptable, while SCP enriched cereal bars and pasta receive positive feedback. Arthrospira platensis in food is well received for its nutritional value without altering taste acceptance. So, what are the economic and environmental considerations? We can examine these from the advantages it offers and also the disadvantages that comes along with it. For example, when it comes to the environmental impact, SCP production reduces environmental pollution by utilizing waste and decreasing land and water use compared to traditional sources. From the point of view of economic feasibility, utilizing local byproducts can lower raw material costs and transportation expenses. SCP production generally has a lower environmental impact compared to soybean meal, and therefore it is sustainable, but it has certain disadvantages like high energy costs. SCP production may require significant thermal and electrical energy, increasing production costs. These figure we see sustainability showing concentric circles with the environment as the central focus based on the hard sustainability perspective. So, given the more number of advantages we have and the less disadvantage like high energy costs, we can think of using alternative energy sources for making SCP production much more viable and sustainable. There are other challenges of using SCP apart from these high energy costs, like high nucleic acid content, which we have already discussed, increases serum uric acid, potentially causes kidney stone. There can be digestibility issues like non-digestible solvents in the SCP affect simple stomach animals and birds. Live microbes must be inactivated to prevent gastrointestinal problems. There is always a risk of contamination. Fungi grow quickly but are prone to contamination. Bacterial SCP has high RNA contamination and endotoxin risks. Certain risks from toxins like mycotoxins, cyanotoxins, carcinogens may develop during production. Algae is toxin free but grows slowly. And mitigation like optimize fermentation, select appropriate microorganisms and treat SCP to reduce anti-nutritional factors. So, some of the emerging technologies for SCP production include various innovative steps like using genetic engineering or synthetic biology or metabolic engineering, which will optimize microbial metabolic pathways to enhance SCP production. Then we can go for phototrophic cultivation, which utilizes microalgae or cyanobacteria to convert sunlight and carbon dioxide into biomass, offering a sustainable approach with high growth rates and simultaneous production of valuable compounds. And it also offers solutions like carbon dioxide sequestration, which is a challenge in the current climate change era. Some of the future prospects are related to sustainability and development, particularly our focus on circular economy, where SCP production can contribute to sustainable development by revalorizing residues for aquaculture and other uses, and also using the waste and then contribute to the circular economy. Then SCP can help address undernutrition due to its high protein, vitamin and lipid content, and it can reduce reliance on traditional protein sources and utilizes readily available raw materials, contributing to a more sustainable food systems. So, here we can see a figure where we see some of the obstacles and some of the future developments in adopting microbial foods, particularly from the point of view of SCP. We consider the economic viability, which is a obstacle, and then also high RNA content and the consumer acceptance due to various issues, including palatability issues or digestibility issues. And then here we can use these as alternative to animal meat. Then we may have enhanced flavor and odors, and it can give us improved nutrients and health properties. And of course, the growth hormone waste is a solution provider to us, and then we can also use these as a food for difficult to reach areas, and it can be ultimate nutrition food. So, with this we come to end of this lecture. Thank you for your kind attention.